Lab diagnosis of fungal infections. Main points. Various specimens can be taken depending upon the site involved: skin scrapings, nail clippings and hairs, scraping from the mucous membranes, scrapings, crust, aspirated pus, tissue biopsy, then blood, CSF, etc. can be taken in systemic mycosis. Various microscopy techniques are used for diagnosis of fungal infections: potassium hydroxide preparation, that is KOH preparation, gram stain, Indian ink preparation, lactophenol cotton blue or LCB preparation, CALCOFLOUR white stain, methenamine silver stain and periodic acid chip stain. KOH preparation. KOH dissolves keratin and cellular material but does not affect the fungi. Hence, KOH or potassium hydroxide is used at a concentration about 10 to 20% and it is added to the specimen and covered with a cover slip and left for 20 minutes in the incubator at 37 degrees Celsius to digest the keratin. Then it is examined microscopically. All the tissue material gets dissolved and only the fungi that they will remain intact. And in microscopy KOH preparation, it will look something like this, that all the tissue material has dissolved but the filamentous forms of fungi can be seen here in the microscopic preparation. So KOH preparation is used generally initially from the specimen, direct specimen microscopy. Directly from the specimen you can do microscopy in the form of KOH preparation and see if fungi are present in the specimen or not. Then gram stain can be performed. Gram stain can be performed from the specimen. It can also be performed from the culture. In gram stain, yeast cells, pseudohyphae, all these structures, they appear as gram positive structures. Indian ink preparation can be used for capsulated fungus known as Cryptococcus neoformans. Indian ink preparation is used for the demonstration of capsule of Cryptococcus neoformans. CALCOFLOUR white stain can be used as it is a fluorescent stain used for various fungi. These are some slides which show CALCOFLOUR white stain showing fungal filaments. GOMORI's methenamine silver stain can be used. And periodic acid chip stain can be used. These are various slides showing these stains. Then lactophenol cotton blue preparation can be used. Lactophenol cotton blue preparation is used to prepare preparation from the culture, from the growth of fungi. So lactophenol cotton blue wet mount preparation is the most widely used method of staining and observing fungi and is simple to prepare. The preparation has three components: phenol, lactic acid and cotton blue. The phenol, which kills all organisms; lactic acid, it preserves the fungal structures; and cotton blue, which stains the chitin in the fungal cell walls. So in lactophenol cotton blue preparation, all the structures will appear blue in color, the background will also appear blue in color, and the fungal filaments will also appear blue in color. But you can appreciate the structure, whether they are septate hyphae or aseptate hyphae, whether there are spores, any fungal structure they are there in the growth of culture of the fungi. For culture, sabrodextrose agar is the commonly used culture medium for fungal culture. pH of sabrodextrose agar is 5.6, that is acidic pH. It does not allow bacterial growth. So acidic pH helps to prevent the growth of bacteria, and besides that, several drugs are also added in sabrodextrose agar to prevent the growth of bacteria like chloramphenicol, etc. And cyclohexamide is also added in this, that prevents the growth of saprophytic fungal species. Cultures are incubated at two different temperatures. One tube is at 25 degrees Celsius, that is room temperature, and one tube at 37 degrees Celsius, that is incubator at the 37 degrees Celsius. This helps to reveal fungal dimorphism. So for systemic mycosis, like for some fungi which have both filamentous and yeast form, in such cases, if you keep culture at two different temperatures, you are able to figure out whether it is a dimorphic fungus or not. Cultures are incubated for at least 2 to 3 weeks, and in some cases it may take even up to 6 weeks for growth, because fungi are slowly growing organisms. Certain fungi like Candida and several species of Aspergillus, they can grow in just a few days, but there are several fungi like dermatophytes, they may take several weeks to grow. Cultures are examined macroscopically for colony morphology and microscopically for fungal morphology. Besides that, Sepex tox agar and cornmeal agar are other culture medias which are useful for fungi. Sabrodextrose agar showing a fungal colony. You can use test tubes or such containers for growing fungi. Like fungi, they may take several weeks to grow, so it is better to grow fungi in such bottled containers, so that moisture remains preserved in the bottled containers and the medium does not dry out very fast. In the petri dish dries out fast, so it is not possible to keep the petri dish for several weeks. So preferably bottled containers are used for growing fungi, so that fungi which are slowly growing, they can be figured out in such containers. Sabrodextrose agar commonly uses for fungal isolation, showing colonies of fungi here. And nutritionally deficient medium known as cornmeal agar, it can be used for growing fungi, and in that spore formation in the fungi can be figured out in such medium which is nutritionally deficient. Fungi will form spores in such nutritionally deficient medium. Candida spore formation in cornmeal agar. Sepex tox agar is also useful for growing fungi. Here it shows colonies of Aspergillus in Sepex tox agar. That completes an overview of lab diagnosis of fungal infections. Thank you.